In this video, I'm going to focus on terpenes and terpenoids in cannabis with a specific focus on hemp-derived CBD products. But before I do, I should disclose that I have consulted for a variety of different manufacturers of cannabis products, and I'm on the medical advisory board of CV Sciences, a manufacturer of hemp-derived CBD products. This is a little bit about me. In most conversations about cannabis, the term terpenes or terpenoids is quickly followed by the term synergy, which is quickly followed by the phrase the entourage effect. But what is the entourage effect? And what does it have to do with terpenes and terpenoids? And how important are these compounds in mediating the effects of products derived from cannabis, including hemp-derived CBD products? I'll do my best to answer these questions. In popular culture and in scientific literature, the entourage effect has become the preferred phrase for describing the synergies created by various constituents within the cannabis plant. This phenomenon may explain how a product containing THC and CBD and a broad range of other phytocannabinoids, as well as terpenes and terpenoids, may have different effects than products that are highly purified and contain only isolated CBD or THC. In botanical medicine, the concept of synergy between the constituents in a plant is not a new concept, but in cannabis it has a catchy new name. Interestingly, the original concept of the entourage effect described interactions between endocannabinoids. It wasn't until later that it was extrapolated to constituents in the plant. The cultural fascination with the entourage effect is well ahead of the scientific understanding of it, especially the clinical significance. In addition to being credited as being important mediators of the entourage effect, terpenes and terpenoids are also thought to differentiate sativa varieties of cannabis from indica varieties. As the story goes, sativas are said to be energetic and uplifting and therefore good to use during the day, and indicas are said to be sedative and analgesic or pain-reducing, and therefore good to use at night. Unfortunately, these cultural narratives of sativas and indicas are not supported by scientific evidence, at least not yet. Cannabis flower and products that are labeled as sativa don't have a consistent terpene or terpenoid profile as compared to those labeled as indica. There is a wide range of terpenes and terpenoids in just the sativa category. Despite this, many people, including Dr. Ethan Russo, have concluded that the supposed differences that are reported by individuals using sativas and using indicas are due to the terpene and terpenoid profile. But I think the differences, if they do in fact exist, could also be due to other things, other factors, like differences in the phytocannabinoid profile, or perhaps the amount of CBD, as well as factors like genetic differences between individuals, which may manifest as important differences in their endocannabinoid systems. Dr. Russo is a neurologist by training, and he is one of the foremost scientific experts on the medical use of cannabis. In this 2016 interview, Dr. Russo states that the sedation of so-called indica strains is falsely attributed to CBD content when in fact CBD is stimulating in low and moderate doses. He goes on to say that rather the sedation in most common cannabis strains is attributable to their myrcene content. Myrcene is a monoterpene, and in contrast to that, a high limonene content will be uplifting on mood. And then finally, alpha-pinene can effectively reduce or eliminate the short-term memory impairments that are classically associated with using THC. Now, it's important to understand that these statements are largely extrapolated from preclinical studies conducted in mice. I'm not aware of a human clinical trial that shows that a cannabis product or cannabis flower that was high in alpha-pinene, for example, led to less short-term memory impairments than cannabis products or cannabis flower that was low in alpha-pinene. Nonetheless, this is an exciting area of research going forward. The aroma and flavor of cannabis, or any plant for that matter, is a function of its essential oils. Terpenes and terpenoids are the predominant constituents in plant essential oils. Essential oils are mostly comprised of combinations of monoterpenes and sesquiterpenes, and we'll talk about the differences between those two. These compounds are responsible for the diesel pininess of some cannabis varieties, as well as the tropical fruitiness of others. And it's been argued that the purpose of terpenes and terpenoids is to protect the plant from damage from microorganisms, insects, or animals, and to also act as a draw for pollinators. All of the terpenes and terpenoids discussed in this video are generally recognized as safe as food additives, according to the US FDA or some other regulatory body, but it's important to understand that their safety is tied to their intended use, which is as a food additives. It doesn't necessarily mean that they're safe to vape, and so some companies will add high concentrations of terpenes and terpenoids to their vape cartridges and then claim that they're safe because these compounds have been generally recognized as safe, but they may not be safe to be heated in high concentrations. Now we're going to talk a little bit about chemistry. So terpenes and terpenoids are lipids. They're composed of repeating 5-carbon units called isoprene units. Terpenoids are similar to terpenes, but they have atoms other than carbon and hydrogen, most often oxygen. The two terms are used interchangeably by a lot of people. And these molecules are very small with low molecular weights, and that means they can convert from a liquid to a gas at low temperatures, including ambient air. Terpenes and terpenoids are classified by the number of carbon atoms and the number of isoprene units. A monoterpene, for example, contains 10 carbons and is composed of 2 isoprene units. A sesquiterpene, on the other hand, contains 15 carbons and is composed of 3 isoprene units, and so on and so forth. Monoterpenes are more volatile than sesquiterpenes. Volatile refers to their ability to escape from a liquid to a gas at low temperatures, and monoterpenes only have 2 isoprene units, and so they're smaller molecules with a lower molecular weight, and therefore they're more likely to escape in the air at low temperatures. And that's why when we open a bottle of essential oils and you start to smell the aromas, you are smelling terpenes and terpenoids in that moment that have escaped into a gas phase. Terpenes and terpenoids may be acyclic, monocyclic, or polycyclic. Acyclic means they have no rings in their chemical structure, like myrcene here on the left. Monocyclic means they have one ring, like limonene here on the right. And polycyclic means they have multiple rings. Over 200 different terpenes and terpenoids have been identified in the cannabis plant. Most current cultivars contain between 20 and 40 of them, generally in very small concentrations, typically below 1% by dry weight. Most of the terpenes found in the plant are monoterpenes. This particular study of indoor-grown marijuana found that over 90% of the terpenes in this sample were monoterpenes, with myrcene being the most concentrated. When looking at studies like this, it's important to note what is being tested. Fresh flower or dried flower or an extract is likely to have a different terpene and terpenoid profile. Given their volatile nature, these compounds can also be lost during the curing and drying process, and that's why there may be a lower concentration in dried cannabis flower as opposed to fresh flower. And given that monoterpenes are smaller and weigh less than sesquiterpenes, for example, they are likely to be lost first. This study of 33 different varieties of cannabis reported similar results. Limonene, myrcene, and pinene were the most concentrated monoterpenes, while caryophyllene and humulene were the most concentrated sesquiterpenes. And this study used dried flower as the samples purchased at dispensaries in Canada. As you're probably aware, different extraction methods produce different extracts. Ethanol extraction and steam distillation typically preserve terpenes and terpenoids better than CO2 extraction, but it really depends on how you do it. If your goal is to retain the terpenes and terpenoids, you can certainly do that using CO2. It really depends on what you want in your extract. Here are 2 certificates of analyses from CV Science's CBD oil. On the left is the raw oil and on the right is the decarboxylated oil. You can see that the raw oil has a higher percentage of terpenes and terpenoids, and this is because some of them are lost during decarboxylation. Decarboxylation is a process that uses heat to remove the carboxylic acid from the natural forms of phytocannabinoids. So ironically, the plant doesn't actually produce CBD and THC, it produces CBDA and THCA, which are forms that include a carboxylic acid. So decarboxylation uses heat to remove that acid to make these molecules more active, at least with regard to THC. And doing so, the heat may cause some of the terpenes and terpenoids to be lost. Here is a certificate of analysis of a hemp variety of cannabis grown outdoors in Oregon. The tested sample was an extract. And if you look at the percentage of THC by dry weight in the extract, you'll see that it just barely exceeds the 0.03% threshold. And this is the threshold set forth in the Farm Bill that determines whether that cannabis or the product derived from it is marijuana or hemp. If it contains more than 0.03% THC, then it's considered marijuana. So the plant material here must have been hemp because by definition an extract is a concentrate, and the concentrate is just barely over that 0.03%. So this product or this extract here might be considered marijuana, and then if you were to dilute this with a carrier oil like MCT oil or olive oil, that percentage would drop below 0.03%, and this would go back to being hemp. So this is very confusing. But the Farm Bill is an agricultural bill, and it refers to the plant material. It's not clear that it refers to products derived from it. Here is the terpene and terpenoid profile of that same sample. The total concentration of terpenes is less than 1/10 of 1%. So probably with this extract, they weren't trying to get a very terpene and terpenoid rich extract. Maybe they were just going for the CBD here. Here is the terpene and terpenoid profile of a batch of CV Sciences plus CBD oil gold formula. There are 5 terpenes and terpenoids that exceed the level of detection, and the total concentration is less than 1%, but more than twice what we saw in the previous certificate of analysis. As you can see, the top 5 terpenes or terpenoids are beta-caryophyllene, trans-neuralidol, alpha-humulene, linalool, and caryophyllene oxide. So this begs the question, how much is enough? How much of a given terpene or terpenoid is enough to mediate a specific effect? Well, Ethan Rousseau, again, has been quoted as saying you don't need very much. Concentrations above 0.05% can have a specific effect, and that's 500 parts per billion, which is very, very small. So by that logic, if we were to simply generalize it, we could see that beta-caryophyllene in the plus CBD oil certificate of analysis exceeds that threshold. But I think what Dr. Rousseau was really referring to is inhalation as a route of administration, because there are very important differences in the way that you introduce these compounds into the body. Inhaling terpenes and terpenoids exposes them to our olfactory system in our nasal passages. They can bind to receptors there and send signals directly to the brain. They can also be absorbed into the bloodstream through capillaries in our lungs. Oral administration, which mostly ends up being ingestion, is different. It takes time for these compounds to be absorbed through the gastrointestinal tract. They're often metabolized extensively in the liver before being released into general circulation. Animal studies in which mice are force-fed terpenes and terpenoids use milligram per kilogram dosages, which are much higher than the doses used in the inhalation chamber. If we look to the world of dietary supplements, we'll see that the doses of terpenes and terpenoids used for a therapeutic effect are much higher. Here are 2 natural products which are composed of terpenes. On the left, we have a product that is basically linalool that is sold and marketed as a dietary supplement and often used to treat anxiety. A single dose is 80 milligrams, which is primarily linalool, a terpene. On the right, we have a product that is peppermint oil, which is basically menthol. It's used to treat IBS, abdominal distension, and bloating. A single dose is 0.2 mLs of peppermint oil, and I don't know the milligram dose of menthol in that. Some companies are adding high concentrations of terpenes and terpenoids to their products and marketing specific effects. In doing so, they're creating these Frankenstein-like products, not close to anything that is created in nature. This is happening in both the marijuana and the hemp industries. It's not entirely clear that this is a good strategy. I'd like to spend some time on these 2 compounds. Myrcene or beta-myrcene on the left is a monoterpene. You can see it has 10 carbons in its chemical formula. And beta-caryophyllene, which is a sesquiterpenoid, you can see it has 15 carbons in the molecular formula, which makes it sesqua. And it's a terpenoid because it has a compound other than carbon and hydrogen in its chemical structure. Myrcene is found in high concentrations in mango, and beta-caryophyllene is found in high concentrations in black pepper. Now, you've heard about these two compounds a few different times during this video. You've heard Ethan Russo say in that interview that indica varieties are sedating because of the beta-myrcene content, not the CBD content. It was also one of the most concentrated terpenes in that one study that looked at 33 different varieties of cannabis. And beta-caryophyllene was also one of the most concentrated terpenoids in that study, and it was the most concentrated compound in the gold formula produced by CV Sciences. Beta-myrcene has been shown to act like a sedative in animal models. It has also been shown to reduce pain and inflammation. It appears to reduce pain through interactions with at least one opioid receptor, and it appears to reduce inflammation by inhibiting the synthesis of prostaglandin E2, which is an inflammatory prostaglandin. One preclinical study found that 10 milligrams per kilogram orally reduced pain behavior for longer than morphine. As a reference point, 10 milligrams per kilogram for the average female in the US who weighs just over 170 pounds according to the CDC. When converted to the human equivalent dose is 63 milligrams, and 5 milligrams per kilogram would be just over 30 milligrams. Another study published in 2016 found that doses of 5 and 10 milligrams per kilogram of myrcene reduced pain in an experimental mouse model. A characterization of beta-caryophyllene, on the other hand, requires a brief discussion of the endocannabinoid system, and that's because beta-caryophyllene is a cannabinoid. A simplified description of the endocannabinoid system includes cannabinoid receptors, the endocannabinoids themselves, and then the enzymes that both biosynthesize and hydrolyze or degrade endocannabinoids. There are two cannabinoid receptors that have been identified to date, CB1 and CB2. To simplify, the geographic distribution in the body is often described as CB1 receptors being primarily found in the central nervous system, and CB2 receptors primarily being found in the periphery of the body on immune cells and on organ tissues. Endocannabinoids are fatty acid neurotransmitters when they're in the central nervous system, and when they're outside the nervous system, they're just lipid signaling molecules like prostaglandins. They are derived from arachidonic acid. Two of them have been identified to date, two primary ones. Arachidonyl ethanolamine, or AEA, also known as anandamide. Ananda is the Sanskrit word for bliss. And 2-arachidonyl glycerol, or 2-AG. The endocannabinoid system is often described as a neuroregulatory system. In this model, we have two nerves that are talking to one another across a synapse. And here, in this particular example, we'll call this glutamate, which is an excitatory neurotransmitter. It's produced ahead of time and stored in vesicles. When a signal comes down this nerve, it spills into the synapse and diffuses to this postsynaptic neuron where it binds to a receptor and activates this nerve. And in response, endocannabinoids are synthesized in this neuron on demand, and they diffuse in this retrograde direction to bind to cannabinoid receptors here on the presynaptic neuron, and this is a CB1 receptor. This leads to a reduction in the production and secretion of this neurotransmitter, which is glutamate in this example. So this is how it can act as a shutoff switch in the central nervous system, or in the peripheral nervous system for that matter. Here is a model of immune regulation. This is a cross-section of the gut, and here is the lumen where food and water would be, and these are the different layers. You can see that white blood cells have infiltrated the wall of the gut, a mast cell here and a macrophage. They're blown up here in the diagram on the right. And so these cells are producing inflammatory chemicals. They're also producing anandamide here. So anandamide is diffusing and binding to CB1 and CB2, primarily CB2 receptors on this macrophage itself, and then also diffusing over to the mast cell and binding to receptors there. And this causes a decrease in the production and secretion of these inflammatory chemicals. So this is the same type of mechanism. It's a shutoff switch. Before we looked at neuroregulation, and this is immune regulation, or more specifically, anti-inflammation. So beta-caryophyllene does this. It has been shown to reduce pain and inflammation via the CB2 receptor in animal models, including doses less than 5 milligrams per kilogram. In 2004, some of the same researchers found that the anti-inflammatory and analgesic effects were positively correlated with the concentration of CB2 receptors. So in summary, terpenes and terpenoids are everywhere in nature. Over 200 of them have been reported in cannabis, but most varieties only contain between 20 and 40. Monoterpenes predominate, but processing and manufacturing can alter the terpene and terpenoid profile. It's not clear how much is needed to achieve a terpene or terpenoid specific effect, and it largely depends on the route of administration. The dose is going to need to be a lot higher if it's ingested. And these compounds participate in the entourage effect, but scientists are still trying to figure out exactly how. Beta-myrcene can act like a sedative and may reduce pain and inflammation. Beta-caryophyllene acts like a cannabinoid and may also do the same thing. With that, I'd like to thank you for your time and attention. Please send me an email if you have any questions.